Okay. Yeah, good afternoon. My name's Haley Oakland, and I'm joined by my colleagues Stephanie Kroll and Amanda Chan from the Academy of Natural Sciences. So welcome to our webinar describing our study, Past Restoration, Success or Failure, funded by the Growing Greener Grant and the William Penn Foundation. The study was initiated to better understand the longer-term outcomes of stream restoration practices take place throughout the Delaware watershed. So while there are many projects being put into place, there's still a lot to learn about longer-term ecological outcomes of stream restoration. Stream restoration projects are usually monitored on shorter timescales relative to the amount of time different biotic communities may take to respond to restoration, if monitoring is done at all. Therefore, we wanted to initiate a study that would look at stream restoration projects in place for at least 5 years to understand what is happening in ecosystems at those sites. So those are our scientific objectives, but we also wanted to work closely with project practitioners within and outside of the Delaware River Watershed Initiative to understand priorities for restoration, practices that can address those priorities, and how to track outcomes. So an important goal of this study was to work closely with practitioners, managers, and decision makers to maximize the benefits to them from a study like this. So to initiate our work, we met with Southeast Pennsylvania County Conservation Districts to understand their priorities. We wanted to better understand what kind of questions they have about ecological restoration outcomes. What kinds of practices are commonly used in their counties? Were there particular project sites they knew of that seemed particularly successful or maybe even particularly unsuccessful? And were there practitioners they commonly worked with that we should reach out to for more information about specific projects? As I'm sure many of you know, stream restoration doesn't mean one thing from place to place. There are many goals and stressors that lead to the use of stream restoration and many practices that fit under its umbrella. This diagram lists just some of the common reasons and practices used in this region. As we discuss priorities and details with both the county conservation districts and with project practitioners, we reviewed these motivations and associated techniques commonly used to address those reasons and tried to narrow down our focus to sites that would be both studyable scientifically and useful for project practitioners. Those initial conversations yielded a long list of potential sites. So this is a picture of Amanda Chan in front of our initial list of sites to review. The sites were compiled lists of sites from the county conservation districts, managers, and practitioners we spoke with. The sites represented several different types of restoration, completed up to 15 years prior to our study. However, a common problem we ran into was a lack of clear information about what was actually implemented and precisely where versus what was proposed in project plans. We needed this kind of information in order to find sites that might be similar to one another. So part of our process of narrowing down sites was also pursuing projects that were well documented. So we focused on those projects that were well documented and that overlapped with restoration questions and commonly used practices of local managers. Therefore, we narrowed down the practices we would study to a combination of 2 or more of the following practices: buffer creation or enhancement, bank stabilization, in-stream structure placement, and channel and/or bank regrading. We didn't necessarily expect these kind of practices to be the most effective ecologically, especially because the projects are often relatively small relative to the scale of degradation and catchment. But we thought these were useful because these were useful practices to study because these are the kind of projects being put into place in this region and also the kind of projects we still have many questions about ecologically. So this left us with a list of over 50 potential study sites. This included some sites from a previous study at ANS that had similar goals. So we started a Tier 1 assessment of sites to better assess their feasibility and compatibility for inclusion in our study. So the yellow sites on this map are Tier 1 assessment only, and the green sites were assessed in both Tier 1 and Tier 2 of our study. During our Tier 1 assessment, we visited sites, oftentimes with the project practitioners, as shown in these images. During the site visits, we wanted to understand whether project components were still visible and intact, and better understand what was done where and why, and logistically whether the site was feasible for our study, such as whether there was a suitable restoration reach and an upstream unrestored reach that could be studied for biotic communities. So we ended up narrowing down our Tier 2 assessment to 17 sites. These sites represented a range of catchment size and land use, from smaller developed drainages down closer to Philly to larger agricultural drainages near the Lehigh Valley. Interestingly though, forested land use did not necessarily track along this gradient, as we had some sites with catchments that were a mix of highly forested and highly developed, or highly forested and highly agricultural land. So now I'll switch over to my colleague Amanda Chang to discuss some of our methods.
Okay, so is restoration working ecologically? So the way we wanted to assess this question is to see how biological and environmental variables would respond to the restoration on both a spatial and temporal scale. The biological communities we chose to look at are algae, specifically diatoms, macroinvertebrates, and fish. So in relation to the direction of the arrow, the order of communities reflects the response of these communities at increasing temporal and spatial scales. So for example, with algae being at the far left, they are the quickest to respond and indicate changes of nutrients, stormwater, and chemical pollution. Macroinvertebrates are next up, indicating changes in local habitat and pollution, and fish communities are at the higher end of these scales and indicate changes in habitat and watershed conditions. All right, so in looking to measure the responses of these communities, we were looking to observe notable or large changes at these temporal and spatial scales. So to do so, our methods included several comprehensive habitat assessments, as well as quantitative sampling of these organisms to analyze their IBIs and other metrics. These methods were done within the restored area as well as the unrestored area to provide a contrast in ecological health. So for macroinvertebrates, 2 composite samples were collected. One composite sample consisted of 3 random Cerber subsamples. The other sample consisted of 5 kickneck subsamples at 1 transect. Depth and velocity readings were recorded at each subsample. The location of these subsamples were within a riffle and had to meet a velocity range of 0.25 to 0.8 meters per second. Next slide. So for diatoms, a 100-meter reach was set and divided into 11 transects that were 10 meters apart from each other. At each transect, a grad sample or substrate was randomly selected and scrubbed to create a composite sample. We also did a qualitative collection of macroalgae, or algae you can see with the naked eye. An example is a picture at the bottom, like water net. That's what you can see. So for fish, like algae, a 100-meter reach was set, for fish, it was also electroshocked using a multi-pass depletion method. This reach was blocked off with nets at the 0 and 100-meter mark, and then starting at 100, we electroshocked the reach moving upstream towards the 100-meter mark and netting any fish we find. After each pass, the fish that were collected were ID'd and then released outside of the reach. We do this 2 to 3 times depending on the contrast of species diversity and species count of the first and second pass. Other organisms that were collected are amphibians like salamanders and reptiles. So for chemistry, we looked at in-stream water chemistry parameters such as DO, pH, temperature, and conductivity using a YSI probe. We did this for fish, macroinvertebrates, and algae. We also collected a 4-liter water sample when algae sampling, just to look at the analytes and nutrients like TSS and nitrates and other major ions, since there's a really strong correlation between algae and these analytes and nutrients. And then habitat assessments. So for habitat, we conducted 4 riparian and in-stream habitat assessments at both a transect and site level. For transect level assessment, we used a survey adapted from the EPA's Wateable Stream Assessment. Like algae, we did a 100-meter reach divided into 11 transects, 10 meters apart from each other. And then at each transect, we looked at in-stream habitat for stream and aquatic organisms, as well as riparian vegetation and any human influence on the vegetation. The site level assessments came from a variety of sources. The high-low gradient assessment was also taken from the weightable streams assessment, and just looked at the quality of the streams, the stream habitat as either a low or high gradient stream, and that depends on what the stream qualifies as. The last two, the QBR index and the RQI, are riparian forest assessments adapted from sources in Spain, but they allowed us to look at the riparian habitat at a spatial and temporal scale. And then on to Steph.
Thank you, Amanda and Haley, for setting that up. So I just want to start by going over what we saw for the habitat. And so this is just a list in alphabetical order of the sites. The R is for restored and the U is for upstream. And then because we can't expect you to make sense of all those bar graphs down below, we just have them, the negative sign is if there was a worse condition at the restored site than upstream, and positive if there was a better condition in the restored site than upstream, and 0 was that there was no change. For fine substrate, of course, that means a better result is it's lower in fine substrate amounts. So we saw habitat respond positively in a lot of the sites, but not all of them. But in all sites, we saw at least one of these substrate variables, either cobble gravel, large substrate like boulders, or fine substrate improved. The pairwise tests were not significant, but we can see these trends. Next slide. For macroinvertebrates, we collected them for 2 years, whereas we collected diatoms and fish only for 1 year, just because of the constraints of the study. We did see the largest difference in macroinvertebrates due to interannual differences. And so for subsequent analyses, we averaged the 2 years of data. So that's what this is. Yeah, thanks. So we're trying to find out, which variables are affecting our ability to measure success. And a strong hypothesis we had was whether or not the watershed was very highly developed. And so development is here on the right-hand y-axis, and these triangles show you the proportion. You can see we've got some highly developed streams. And then the color coding of these bars is the greens show an improvement in the restored reach. The yellow shows, not a really large improvement, just 1% difference. And then the orange and red show that the upstream not restored reach had a higher value for the Pennsylvania DEP's IBI score. And so when you look for the green across this gradient of development, there actually is not a strong pattern showing that there's more success in less developed areas, which is what we would assume. But at the same time, this is encouraging to see that we could see some improvement in these streams, even though there was high development, so that IBI scores, or this index of biological integrity that talks about the macroinvertebrate community health, is responding positively to the restoration, even in more urban areas. Next slide, please. So for the fish index of biotic integrity, we saw improvement in more places than not. And so that was encouraging because the fish can show like a long-term, more significant improvement in a stream. Next slide. And for fish, we saw direct relationships with some of the habitat variables. And so cold water species were associated with more riffle and smaller streams. So this is greater wetted width, or a wider stream. And down here, the coarseness of the substrate was associated with certain fish species. So this is helpful in considering how these individual pieces that are related to stream restoration can be related to fish species directly. Next slide. When we looked at algae, in this plot, it's showing the difference between the unrestored or upstream site and the restored site. So we are seeing differences in diatom communities between the unrestored and the restored sites. And then we've got a grouping of the more developed sites here and the more agricultural sites here. But we're not seeing a clear pattern of direction. So we're not seeing all of them go from one place to improved in the same direction. And we think that these practices that were put in were mainly to stabilize banks, to reduce temperature. They could affect light if the riparian zone had grown in. But other than that, they were not targeted specifically to improving diatom species and algae growth. So we weren't too surprised, since algae do tend to respond more closely to stormwater events and nutrients, that they aren't showing a lot of change based on these projects. And so just to look at, this is basically a measure of how different the two reaches, the upstream reference and the downstream restored, are for the three different biological indicators: algae, macroinvertebrates, and fish communities. And so you see, as we saw before, a lot of differences in the fish community, which is encouraging that that restored reach, which isn't that far from upstream unrestored reach, is showing a pretty good difference in the fish community. And then we're seeing moderate differences for the macroinvertebrate communities and algae communities. Next slide. And so if we just put some of these out for some of the more significant projects, you know, we didn't have huge expectations. As Haley mentioned, the scale of these restorations tends to be pretty small, a few hundred feet to, well, yeah, about that, a few hundred feet, in reference to these watersheds, which could be quite large. So even though sometimes it looks like a lot of negatives, we did find it very encouraging to see all the positives here and all of the change observed. And it might not be consistent across every category, but just to see that, you know, 7 of the sites had an improvement in macroinvertebrate IBI, 5 of them had an improvement in fish IBI, fine sediment improved in many, as did cobble gravel, and the overall habitat score did. I don't think we got, because the sites, well, we only had 17 sites, even though here we have fewer listed. And because they were so different, we didn't find a lot of concrete answers as far as like what is really influencing their success. And so that's a question that I think a lot of stream restoration researchers are asking. And it definitely requires a study that is a little bit more intensive than ours. Next slide, Haley. Thanks. But I did pull out like the successful projects, the one where we did see a change, and then rerun an analysis just to see what did we see change and what relationships did we see in these projects. So we saw that those macroinvertebrates that filter feed responded to boulder, cobble gravel, fine substrate, chloride concentration, soluble reactive phosphorus, and they also showed a relationship with watershed size. Taxa richness, or the number of different macroinvertebrate taxa we found, were related to all of these variables that I've presented. Of course, we had analyzed a much longer list, so I'm just summarizing the ones that showed these relationships. IBI score was closely tied to fine substrate, chloride, watershed size, and bank vegetation. And then EPT richness is, sometimes people act like it's the magic indicator, but I think it's always worth considering multiple metrics, because here we see, well, we know we don't get a whole lot of stoneflies and sensitive mayflies in these areas. So in these suburban and urban streams, EPT family richness, it may not be the best indicator of response, but it still responded to chloride, phosphorus, and watershed size. Next slide. And so in this portion of the study, you know, we were really encouraged to see that even in some of the high development watersheds, there were improvements in the biotic communities. And whenever cobble and gravel was introduced as part of in-stream habitat restoration, it seems to be effective in bringing better, you know, healthier biotic communities. And then especially cold water fish assemblages did respond to widening of the stream, which can happen with riparian restoration, as well as improved riffle habitat. And I believe I now hand it back to Haley.
Always got to remember to unmute. Okay. Yeah, thank you, Steph. So now I'll briefly review a portion of my master's research, which was built off of the study we've been discussing. So my research was funded under the Maryland Sea Grants Coastal Resilience and Sustainability Fellowship. And for my research, I wanted to focus on physical habitat within restored streams. So this is a figure from the National River Restoration Synthesis Study that shows the top 5 objectives of stream and river restorations in 7 major US regions. The green in this figure represents in-stream habitat improvement, and the box shows that the Chesapeake Bay region, encompassing Virginia, Maryland, and Pennsylvania, had the largest proportion of projects with in-stream habitat improvement as a major objective. So if restoration projects are being implemented in this region to improve stream habitat, it's important we're adequately understanding how the habitat is changing to properly understand restoration outcomes. But what constitutes in-stream habitat improvement? So habitat heterogeneity has been long suggested to be important to ecology and connected to biotic community diversity. Within the restoration ecology community, as shown with this quote, many have suggested that ecological restoration projects must account for physical heterogeneity, given its influences on key ecosystem processes, functions, and diversity. However, many restoration ecologists also question particularly in terms of variable spatial scales of heterogeneity. But we're rarely able to discuss heterogeneity across more than 1 or 2 spatial scales, because we're often limited by the scale of the field reach or smaller. However, recent developments in surveying and imaging technology may allow us more flexibility in spatial scales of analysis. So for my study, I employed a type of photogrammetry to survey the physical habitat within study streams. The technique uses overlapping photos to produce 3D models, in this case of the landscape. It's similar to stereoscopy, where overlapping imagery viewed through a stereoscope, like this one in the upper right, can be seen in 3D. My method uses a similar concept, but using a computer algorithm. Given the distance between images and the spatial relationships between features of adjacent images, this technique can project points representing the relative 3D orientation of features. This method has experienced a renaissance in many disciplines and has shown a lot of promise in freshwater research. These figures are from researchers who have successfully used these techniques to model physical components of river systems. So given the success and the widely available high-resolution cameras on drones, it seems plausible that this method could be used to assess physical habitat with greater objectivity, spatial extent, continuity, and precision than current field methods. I studied a subset of 3 of the sites from our larger study in Southeast Pennsylvania, in addition to 4 sites in Maryland that employed similar restoration practices and had similar watershed characteristics. I wanted to compare physical habitat heterogeneity between restored and unrestored reaches at my sites to understand whether there was any difference in heterogeneity and at what scale. I started at the scale of the entire restoration project to compare the overall heterogeneity between the entire restored extent and the entire unrestored extents within my study areas. I used approximations of cross-section zones to extract cross-sectional mean values of depth, width, and flow velocity. But I couldn't simply compare all of my restored cross-sections to all of my unrestored cross-sections because the restored extents were often larger than the unrestored extents within my study areas. So I randomly sampled an equal number of cross-sections from both extents. So each of these lines corresponds to one of these cross-section zones from which I sampled mean depths, widths, and velocities. My samples contained 25 cross-sections per extent, and because any of those subsamples may not be perfectly representative of the extents, I repeated this 100 times. So I repeated this process at the scale of the whole restoration project and also on smaller local scales. Locally, I wanted to know if I'm at any given cross-section in the stream, what is the variability within a certain distance away from me? So basically, my methods looked at distributions of values of depth, width, and flow velocity, and compared whether the variation of those distributions was different between restored and unrestored reaches. So I performed the same analysis at the scale of the entire restoration project and at local scales, ranging from 10 meters to 200 meters. The D, W, and V in this column represent depth, width, and velocity for these different length scales of analysis. And the columns are for the different sites I studied. So this table will show colors relative to the results I saw. So white cells mean there was not a major difference in variation between the restored and unrestored reaches for that variable site and length scale combination. Orange meant the restored reach saw greater variation than the unrestored reach at that length scale. And blue meant the opposite, that the unrestored reach had greater variation. So when we look across all the length scales, we start to see that many of the comparisons indicated greater variation in the unrestored reaches, particularly at broader length scales. So looking at the restoration objectives seemed to at least partially reveal what might be contributing to these findings. Qualitatively, it appeared that restoration projects with the objective of mitigating the hydrologic regime were associated with restored habitat that was less heterogeneous than the surrounding unrestored habitat. Alternatively, it seems that the sites that mostly saw the opposite in comparisons, with either no significant differences or with higher restored variation than unrestored variation, were those projects that prioritized aquatic habitat improvement, but not also hydrologic regime control. So I did find many instances where patterns of heterogeneity in physical habitat were significantly different between restored and unrestored extents. These differences were variable based on the scale of analysis. As I said, it seemed that project objectives may be associated with these outcomes, as there were distinct characteristics of analyses associated with restoration objectives. Outcomes are also likely related to other conditions at these sites, like local land use, stream and watershed size, and other characteristics. Overwhelmingly, we saw comparisons that indicated either greater heterogeneity in the unrestored extent or no significant difference. So this indicates a failure in increasing heterogeneity in these restoration projects. If, as I showed from the National River Restoration Synthesis Study, the dominant project objective in this region is aquatic habitat improvement, and if heterogeneity is important to aquatic habitat, restoration objectives. These methods represent an incredibly exciting new way to understand what restoration is really doing to aquatic habitat. Similarly, it might help us to distinguish when the byline of ecological benefit can and should really be added as a potential outcome of restoration. There are numerous interesting ways these analyses can be furthered. The most prudent is to assess whether these outcomes in physical heterogeneity are associated with biotic community outcomes and at what scale. There's considerable skepticism in the restoration ecology community about blindly accepting physical heterogeneity as important to biotic outcomes. So this kind of analysis may help the freshwater science community to tease out some of these ecological interactions. Of course, the physical habitat is only one component of a healthy stream. So you can't just, quote, build it and expect them to come. So it'd be necessary to consider how this physical heterogeneity affects biotic outcomes in the context of other variables like water quality, hydrology, and the existence of biotic community source pools. But ultimately, these methods allow us to analyze variable scales of physical heterogeneity, which may help us tease out its importance in stream ecosystems. So overall, when we look at past restorations and ecological outcomes, we saw a mix of success. As Steph showed, we saw improvements in biotic communities was possible even under relatively high developed catchment conditions. We saw improvements in macroinvertebrate IBI scores and other metrics related to improved substrate conditions. And we saw improvements in fish communities related to improved habitat conditions. In my master's research, I found there were times when restoration objectives may be conflicting and that heterogeneity may not be able to be increased where hydrologic regime control was a major objective. But I did see improved heterogeneity in habitat at sites where habitat improvement was explicitly targeted. Overall, as many of us know, it's likely that successful habitat improvement will only be able to improve biotic conditions if larger catchment stressors are addressed, such as chemistry and hydrology. So we'd like to thank you for joining us today. And now we'd like to first take any questions and then to start a discussion with you all. As we know, we all still have many questions about ecological restoration, and we're still very interested in studying any interesting restoration sites you may know of.
Thanks, Haley. So yeah, if folks have any clarifying questions or any questions at all, you can just, I think you can just unmute yourself, just write in the chat if that doesn't work. I haven't had to host a webinar before today, so there may be glitches.
Hi, Haley and Stephanie. This is Aaron Klauser. I do have a question. Thank you for the really good information and data. My one question is, for a lot of projects, the areas upstream or downstream were not targeted for restoration because they might be a mature forest or better habitat-wise, something like that. So rather than areas not in need of restoration, for Plum Run, I might refer to it more as an area that does not need restoration at this point. How do you control for that when you're looking at this data is sort of my question. Thank you.
Yeah, thanks, Aaron. I know we struggled with the fact that not all upstream reaches were referenced. So for Plum Run and for some other ones, that upstream site was very good. And then for others, the upstream site seemed worse. So we tried to just run our analyses to look for a difference. And then just looking at the data afterwards to see, you know, whether that difference showed that it was much worse or much better. But that is definitely part of the complexity of the study, where we couldn't find consistent sites that had all improved or all healthy upstream areas or all degraded upstream areas. But I think we did just sort of fall back on a lot of the graphs and charts and those ways of looking at the data because the statistics were failing to consider that type of problem. So it ends up, a lot of the results here are really more stronger in an anecdotal sense. But good question. Yeah, in Plum Run, you could see one of them showed that it was, well, I can, well, Haley could back us up to one of those slides, but that was a chain of like 3 sample sites around 2 projects that I believe you have worked on.
Right.
So we did see like a worse downstream effect relative to one of the sites, but then better, yeah, at the second one.
Mm-hmm. Yeah. If we're able to bring those sites up anywhere close to the upstream area, I'll be really pleased with that. So, but yeah, thank you very much. I appreciate it.
Thanks. Wow. I mean, yeah, we can get a drink and hang out. No, and discuss other items. Or yeah, if there are no other questions right now, I know personally I have a hard time on the spot asking questions. So I'm happy to any later. Oh, Scott, I see you have a question.
Hey, Stephanie, while that you have that slide up, did you see any indication that there was a higher likelihood of success by any metric over time with a particular site? In other words, does the length of restoration increase apparent success?
I would love to do that study. I think, unfortunately, a lot of these sites were not monitored before the restoration went in or during the period of time. So these were 5 to 15-year-old projects when we studied them, but we weren't able to study them over time to see. What we were hoping was that enough time had passed where we could see if the change had endured. But I think that question is a big one that we all have as far as the timeline. And then restoration trajectories, you know, can be it improves to its old condition, or it takes a little while and then starts to improve after a lag time, or that, you know, it improves not quite to its, you know, previous condition. So anyway, I wish I had an answer for you, but we're hoping by establishing some of these relationships with the practitioners, we could start studying things closer to when they're put in and do a study like you're asking about.
Okay, thanks. And while you have the slide again, what are these particular sites? What are you emphasizing these for of all of the restoration sites?
This is where we had the most complete data collected. So for example, we didn't collect fish at all of our sites. So these two that are blank, these three that are blank don't have fish in them. And also I think we just left out a few that didn't show as much improvement across these sites, but I'm happy to share. We have some other ways the data are presented that are more complete, as well as a report, and I'm happy to share those out. I think we just didn't want to clog your eyes with too much here. So these were the most successful sites, except for Lesher's Run here, poor thing, which didn't show much change. Thanks. Okay, we have one in the chat from Ellen. Haley, do you want to answer Ellen's chat question?
Sure, let me just pull it up.
So in case anyone can't see it, it says, for clarification between the two studies in terms of big picture takeaways, one seems to indicate benefits from restoration for habitat characteristics, but the other showed mostly a reduction in habitat heterogeneity.
Yeah, so I guess this was, that is a good point for clarification. And I think it was part of why I was interested in studying the physical habitat characteristics, because I think there are a lot of different ways you can dice it in terms of looking at outcomes of restoration, even just in the physical habitat. So a lot of times we'll look at, you know, like the overall EPA habitat score and try to compare restored to unrestored and maybe not see a difference. But I was interested in looking at the heterogeneity to see, even if we didn't see a difference in something like the average depth between the two sites, was there a difference in the heterogeneity? So as I said, I studied sites in Maryland as well, and I was just looking at the physical habitat. So I did see a lot of reduction in habitat heterogeneity within restored streams. But, you know, I also saw some increases in habitat heterogeneity, and I wasn't able to present my results or I wasn't able to analyze differences in substrates and between restored and unrestored. And so that was a big thing that we saw with the larger overall study, was that their restoration, obviously, as a lot of you know, introduces a lot of larger substrates, and that did seem to be associated with some of the better biotic outcomes. So like I said, something I think would be prudent is to understand whether the biotic outcomes are related to the heterogeneity outcomes I saw.
So this is Ellen. Hey, thanks for that. And I'm not a biologist by training, so forgive me, but so would it be accurate then to, like, we know this is very small sample size, blah, 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 right? Like, we shouldn't take big general takeaways from any of this, but is one of the things that we're sort of learning here is that maybe the change, like habitat heterogeneity, isn't a great indicator of whether your restoration project has been successful or not, because it could actually have had some great benefits, even though we might, like, so that changing the physical habitat may not be potentially might not be as important of an indicator as anyway, sorry. That's
yeah, no, that's
okay.
Fine. So I think that's an interesting point. So like I was just saying, it hasn't been compared to the biotic outcomes yet. So but yeah, I think that would be a really important next step, and something that would help to answer that question is if the heterogeneity or lack thereof that I found is related to biotic outcomes, then we see that that's important to be including in our restoration projects. If it's not related, then that's interesting, because that means a lot of our assumptions about heterogeneity at different spatial scales might be wrong for either freshwater systems or just for restoration projects in general. And then, like I said, at the end of the portion talking about the study, I think it's also important to consider in the context of kind of like other watershed characteristics, like is there still stress from chemistry? Has that been addressed? And if that's already been addressed, then maybe the improvement in heterogeneity will have a more benefit and kind of more of a chance of improving the biotic conditions. So unfortunately, I think the answer is that we still have more questions about that.
That's, yeah, perfect. That's very helpful. Thanks, Haley.
Yeah.
And Haley, there's another question in the chat there. I'll just chime in while you're reading that one to say that I think that question about heterogeneity and is it improving the stream condition and the biotic condition and our need to understand that also just highlights the different, like, conflicting motives for the stream restoration. So we had a lot of interest when we started the study in looking at stormwater measures, but we just couldn't find enough of them connected to streams where we could run that study. But I still think the stormwater issues are still deserving of a lot of research in reference to stream restoration. And so that was one of the big results that Haley showed, is that they do reduce heterogeneity, but are they improving impacts of stormwater? And then many other projects are specifically targeting improving habitat for biota. So I think one of the big findings of Haley's work is that you can't necessarily tackle those two at the same time, or these projects may not have successfully tackled them both. And it's worth examining, is it possible, what techniques could accomplish that?
Yeah, and this question from Mark. So I didn't use any other metric than the coefficient of variation. If you have any suggestions, I'd be happy to take them. But I guess, the analyses that I did were a little bit more complicated than the depth that I went into for this talk, but mostly I was looking at the coefficient of variation when I was looking at a distribution of values from the whole restored area and the whole unrestored area. And then I was looking at distributions of values locally. So like I said, like within 10 meters or up to within 200 meters of a given cross section.
Thanks for the question, Mark.
Yeah, and you know, of course, if there are any other questions, please feel free to contact us. And like I said at the end as well, you know, if this has kind of spurred in you any thoughts about interesting sites that you think could be cool to study, please let us know. We'd love to partner with people to look at studying other interesting sites as well.
Stephanie, I'll take one more.
Great.
So do you believe there's data here to start to evaluate which restoration practices specifically are providing value?
I don't think we have, well, I think we found that improving habitat by placement of boulders and cobble and reducing fine substrate were effective. And of course, I guess we just have to say with the caveat that they weren't washed out after the restoration, so they remained there for 5 to 15 years. I know there are plenty of data that show that riparian restoration and bank stabilization are effective. We had hoped initially to design this study so that we had a subset of sites in each category of in-stream improvement, bank stability, bank grading, and riparian forestation. But we did not have enough in each of those categories to run that. So I think, as I said, and you know, Ellen repeated, we appreciated that we saw some change, but we do think that this is a very like pilot preliminary study. So you can talk about it in those instances and use the results as showing that there has been change. But yeah, hopefully we can continue to study it. So yeah, I think the in-stream, just introducing better substrate is showing improvement. Okay, we have Brian Winslow. Are there any plans to continue to study these projects and add any additional sites? So I think we'd like to meet with the county conservation districts and other restoration practitioners to see, as we did when we started this study in 2015, if they have suggestions, and then apply for funding to do so. So I'd say we do have plans, but they're not so concrete. They're still in the thinking phase. But definitely want to hear from you if you, as Haley said, have sites you would like us to consider. And then Brian Vadino says, were there particular trends and systems influenced by limestone geology versus non-limestone geologic areas? We didn't see huge variations in the geology at these sites. I know within the region we do have some limestone sites, but as I remember in that analysis, we didn't find that geology was that different among these sites, so we didn't include it as a covariate in our analysis for our list of environmental variables. But yeah, certainly that can definitely affect groundwater infiltration, the speed with which nutrients come in or out of the stream. Anyway, you know. Molly, yes, we'll definitely be sending out the PDF and the recording of the presentation. I had never set up a Zoom for people to register, so it looks like that was successful. And so we'll send them out. And if we are unsuccessful, you'll get an email. But thanks for being our guinea pigs there. Well, we can just leave a little bit of space here if anyone is thinking of any questions. But also, we'll include our email addresses in the recording and the, sorry, I don't know if I can't speak, the presentation slides. I will say Haley, our wonderful Haley, is moving on to get her PhD in August. So we'll probably give you her new email address rather than her Academy email, even though we'll know how to find her. That way you can also find her. Well, and I'll just tell you, Amanda Chan is working part-time for us while she is in school. So she's been a really strong partner in our field work and this work and the DRWI. And so it's really nice to be able to work on this while she is getting her degree and having her contribute a lot to this study and to our other work. All right, well, we can stay on if anyone wants to hang out, but we really appreciate you all joining in, and good to come together in these times, and you know where to find us. Thank you. Well, thanks for all the chat comments.